Hi, I'm Cassi Niemeyer. I'm going to be a senior in biochemistry at Valparaiso University.
And I'm Alyssa Jarabek. I am going to be a junior chemistry and biology double major at Valpo.
And I'm Natalie Michaels. I'm a junior at DePaul University studying cell biology.
Okay, and we are presenting on the synthesis of an unnatural fluorescent amino acid.
Okay, so to start off, I want to give a little bit of a background on our project. So our most recent attempt at the synthesis used biocatalysis, which is just using enzymes or microbes to synthesize organic molecules. And we're trying to synthesize this unnatural fluorescent amino acid, or UFAA, which is shown above and is closely related to L-tryptophan, which is over here on the left. And we're synthesizing it into this UFAA shown on the right, and this boxed part in orange will be the fluorescent group that will make our amino acid glow. And it's the proposed route for our project uses glutathione, which is shown below, which is actually just a detoxifying agent for the human body, and using glutathione S-transferase, which is an enzyme, to synthesize our UFAA. So here's the overall synthetic scheme for our project. And in the first step here, we're going to be converting the thiol into an amine. And then in the second step, we will use glutathione S-transferase to perform a nucleophilic aromatic substitution reaction, in which we're going to be removing chloride ion from that molecule there and replacing it with our glutathione molecule. And then in the third step, we'll be removing the two side chains from glutathione.
So as Alyssa mentioned, we are going to use glutathione S-transferase to catalyze the second reaction, which is a nucleophilic aromatic substitution. And the one we have chosen is a thermophilic organism, which helps us with the purification process and for thermal stability. And our source is the Thermosynechococcus elongatus, or T. elongatus, which was a blue-green algae found in Japanese hot springs. And this GST has already been proven to work with a very similar reaction and our reaction as well. And so we then optimized the gene to better fit E. coli mechanism. So it just basically optimized the codon sequence. And then we added a his tag to the GST to first simplify the protein purification, and it will also allow enzyme immobilization.
So now we're going to talk a little bit about the results we gathered this summer and what we've been able to accomplish. So as Cassie mentioned, we started with a plasmid construction that included the T. elongatus GST gene. We actually got that from a company called GenScript. And as she mentioned, that included both E. coli codon optimization and a his tag. And then we transformed the E. coli with this plasmid, and the GST was expressed. It was induced using IPTG, and then we purified the protein using a nickel chelate column. Once we got our purified GST, we learned that it was enzymatically active using a standard substrate for nucleophilic aromatic substitution reaction. We learned that by doing a protein assay, and then we were also able to do a Bradford protein assay of also the purified GST. And then finally, a little bit about our future work and where we plan to take the project in the future. So we want to be able to do an SDS page to assess the purity and determine or approximate the molecular mass of our purified GST. We want to do some more plasmid prep and DNA sequencing of the plasmid from our specific expression strain of E. coli. We also, as Alyssa mentioned, want to learn more about how to replace the glutathione thiol with an amine, as we did in step 1 of the synthetic scheme. We also want to keep going and learning more about step 3 in the synthetic scheme, which was taking away those two side chains. So once we get this synthesized, the UFFA will hopefully be passed along to Dr. Rowe and her students, who are then going to attempt to incorporate it into their proteins using their existing tRNA synthesases. So we want to give a shout out to the Pre-Medical Arts Endowment for the funding, the Valpo U Chemistry Department, and all of the alumni who worked on this project in the past. And then here's just one more look at our overall poster. So thank you for watching.